Okay, do you hear me? Is it okay? All right. So yesterday or 2 days ago, we had a talk about the interaction between the host and the pathogens. And we have seen both components and the interaction between both in terms of aggressivity of the pathogen or not, and the immune responses, the types of immune responses. We have been looking about the type of immune responses in confronting each of the different pathogens we deal with, the viruses, bacteria, if you remember, and we have seen some evasion mechanism of the pathogen to the immune responses. So that was the first talk. So today we will go to more in depth of the understanding of the immune system as we know it today. So this is what we call basic immunology. So these are the fundamental elements of the immune responses, and we will go through them, always keeping in mind that the immune system is mainly directed against foreign pathogens, okay, but could also be involved in some other immune processes like autoimmunity, immunity to cancer, allergy, etc. Okay, so let's start with the two major components of the immune system. You remember yesterday we talked about innate immunity or natural immunity, immunity that is there and that will be started immediately when the pathogen enters the body, okay? And there is a second type of immunity. What was it? Adaptive immunity, the specific immunity that will be directed specifically to that single pathogen that has been entering the body at that specific time, okay? So today we will focus on innate immunity. There will be another talk, I think at the beginning of next week by Professor Tabara about the adaptive immunity, okay? So this is just a rapid reminder. Microorganisms are confronted by all the defenses of the body, and I have anticipated yesterday some of them. Remember that we have barriers, physical barrier. Can you remember yesterday we discussed it, and we will go back to that. And we have the innate immune defenses that I'm detailing today, and the adaptive immunity, of course. So when we get infected, there are, as we all know, different outcomes of this infection. Not every infection will lead to disease. Remember, we have been talking about the concept of inapparent infection, okay? So when we get infected, there are different outcomes that could happen. Either the infection is controlled by the innate immunity and nothing happens. There's no disease, it's controlled. When you have a boil at the skin, the polymorphonuclear cells will come by the inflammation process. They come there to the site and they kill the Staphylococcus aureus and they die with it. And that's the pus you have at the boil site. And it's over, okay? There is no more problem. You will heal the skin and that's it. But from time to time, this is not enough, of course. And then either you will have the disease or you will have the adaptive immunity that will start. This is specific immune response. And this could help in most of the cases, fortunately for our species, to recover from the disease. So first step, potentially different second step. There's another aspect which is important in terms of immune response. It's when you get reinfected by a pathogen that you have encountered first, okay? So this is very important. And this is mainly adaptive immunity will be involved. Since adaptive immunity, when you have a response to the pathogen, we will produce lymphocytes B and T against the pathogen. Most of them, when they finish their activity against the pathogen, will disappear. They have a half-life. They are not anymore there. But some of them will come back to a resting state, and these will be the memory of the pathogens. This is exactly what happens when we vaccinate people. We give them first dose of the antigen, and then we come back with several other doses of antigen. The idea is to develop this memory against that pathogen, that specific one. And then we protect them this way. So these are the different figures and outcomes possible of the infection. Now, today we will focus on the innate immunity, as I said. You all know that we have, for most infectious agents will enter either by the skin, if there is a lesion in the skin, or if there is a vector that comes and injects the pathogen through the skin, which are relatively rare situations, but most of them will enter through the mucosa, of course. Yeah? The mucosal tissues are the most important point of entry of pathogens. When we breathe, when we eat, when we have sexual contact, those are the places or the sites where the pathogen could enter the most frequently. So on those, either the skin or the other mucosa, we will have a number of innate immunity defenses which are there. We will have enzymes in all of the secretions, in the eyes, in the ears, in all mucosa, in the bronchi, of course, etc. We have in the bronchi we have the mucus and the cilia, which help eliminate the pathogen to get it outside. The same thing in the gut. We have the acid in the stomach itself, but we have also, I've discussed it yesterday, the commensals, all that microbiota that is there in our mucosa and that creates an equilibrium with the pathogens. Okay, remember we discussed that aspect yesterday. So we will have at the skin also, we will have barrier, the physical barrier itself, which is intact in most time, but sometimes it is not. But there is fatty acids, commensal as well in the skin that could also interact with the pathogen and limit the action of the pathogens. Same thing at the urinary tract, the pH in the vagina, for example. All these are components of our immune defenses. They are there all time. So whenever any pathogen comes, we can already fight the pathogen with those possibilities. That's not always enough, of course. And then we will have to go to further natural defenses in this innate immunity. And the next step are cells like phagocytes, NK cells. I will come back, of course, to each of these and the mechanism of action of each of these. So cellular component, but we have also a soluble protein, so a humoral component in that innate immunity, which include this complement system. I will go into details of this today. Cytokines, which are the molecule which interact between cells, which make the communication between cells, and the interferons, which are a very strong system that help us fight mainly for the interferons type 1, the viruses. Okay? And I will go into some of the details about all these aspects. So now, what are the components of the immune system? I would not enter into detail of the lymphocytes, the specific lymphocyte, the T cell and B cell, that will be done next time. But we have the large granular lymphocytes, which are the NK cells. Morphologically, they are large granular lymphocytes. These are the NK cells. NK cells are lymphocytes. We have three types of lymphocytes, B, T, and NK. Okay? NK are part of the innate immune system. They do not have specific receptor for a specific peptide of the antigen. All right? So, second, we have the phagocytes, which are extremely important. Part of them are the mononuclear phagocytes, which are monocytes in the circulation, and macrophages when they localize in the tissues. We have neutrophils, extremely important in terms of phagocytosis. Okay? When we will see how do they work. And we have eosinophils, which are important in allergy, in responses against ailments, parasites, helminthic parasites. But we also have basophils and mastocysts, which are very important in allergic diseases. Eosinophils as well play a role in allergic diseases. We also have platelets. All of them are very important in as inflammatory mediator production. We also have at the level of the tissue cells these interferons and cytokines which might play a role, of course, in the defense, as I said. So these are the components of the immune system. They will help produce for the specific one antibodies, cytotoxic T cells, but also the complement is produced by mainly by the liver, but we still have some cells, immune cells, which could produce complement locally at the local level. And then you will have the mediator of inflammation, which are very important, and as I said, the interferons. Now, just in terms of morphology of the cells, just that you could keep that in mind, please. We say it more than one time so you know what are the different components. So the neutrophils, we call them polymorphonuclears because their nucleus here, you see the nucleus here, it's not one nucleus, it's polylobular, okay? That's why we call them polymorphonuclear. We can recognize them because of that. The macrophages, which are different morphologically, the size is approximately the same, a little bit less than polynuclears. And then we have the granulocytes. We have the basophils, the eosinophils, and the lymphocytes, of course, specific immune cells. The granulocytes, as you can see, include the basophils, which have these elements in the cytoplasm. These are the enzymes, the granules that produce the different enzymes that are important in inflammation in different allergic responses, as I said. Eosinophil, different in terms of morphology, but also characteristic by the presence of granules. And the lymphocytes, which is the immune or adaptive immunity, which are smaller cells, but with huge nucleus. The nucleus is very important, and there is a very thin cytoplasm. Okay? So these are the white blood cells. Now, the main population of phagocytic cells, we have the mononuclear macrophages. They are formed as monocytes in the bone marrow. They will circulate as monocytes in the blood for 24 hours or something like that. And then they will settle in the tissues and become what we call macrophages. We have polymorphonuclear cells, which are the neutrophils. These two populations constitute the phagocytic cells. They are extremely important, and particularly the macrophage will play not only the role of phagocytosis, but they will also play the role of antigen-presenting cells to the lymphocytes, okay? And they will start in some way the specific immune response or the adaptive immune response. Is that clear, please? If any questions, it's okay now, huh? All right. So, the phagocytosis process. You will have, this is the phagocytic cells, this is the antigen in blue, and there will be non-specific receptor to which the pathogen could adhere. This non-specific receptor could be receptor for some fractions of the complement, could be receptor of the FC fragment of the immunoglobulins that are attached to the bacteria and that will be used to let the bacteria enter the phagocytic cells. So once this happens, this interaction, you have the formation of the phagosome with the part of the membrane, of cytoplasmic membrane of the cell. And within the phagosome, you will have the bacteria and either the immunoglobulin, if it's an immune complex, or fraction of the complement that are inside. Once we are inside the phagocyte cells, this phagosome will interact with the lysosome. Lysosome is an organelle in the cell which has enzymes. Enzymes, which role will be to kill the pathogen. Okay? And I will go into details. So this is what we call the phagolysosome. Okay? Phagosome and lysosome. Together, phagolysosome. So inside that, you have the pathogen and all the machinery to kill it. Okay? When everything is efficient. And as I told you yesterday, if you remember, in the evasion mechanism, one of the evasion mechanisms is to inhibit the fusion of the phagolysosome. That's what you use, for example, mycobacteria. So it's important step. Once we have this phagolysosome, of course, we will release within the phagolysosome a number of microbicidal products which will be produced. And we will see this in this step. We have several mechanisms. Some of them are oxygen independent. They do not need presence of oxygen. And these are including a number of enzymes that are damaging the microbial membranes, the lysosome, which splits the mucopeptide in the bacterial wall, lactoferrin, proteolytic enzymes that digest the peptides of the pathogen. So all of this machinery is available. It's oxygen independent, and within the phagolysosome it will act on the pathogen to kill it. But we have the most potent mechanisms, which are the oxygen dependent mechanisms. These are very important. Here is represented the membrane of the phagolysosome, and in the membrane of the phagolysosome we have an enzyme which is called NADPH oxidase. Extremely important. When you do not have, for genetic defects, NADPH oxidase, there is a very severe disease, which is chronic granulomatous disease, that happens. So this is a very important enzyme. So this enzyme will play a role by a transfer of electrons to produce what we call superoxide ions. They come from oxygen and this transfer of electrons, O2 minus. These are extremely toxic for the pathogens. Okay? Not only that, but they will produce H2O2. What is H2O2? It's peroxides. Peroxide, so extremely toxic as well. And not only that, when there is the intervention of an enzyme called myeloperoxidase, in presence of chlor, we will have HOCl minus. What is HOCl minus? What is hypochlorite? What is hypochlorite? It's bleach. You know the bleach that you use to imagine what is produced within the phagolysosome to kill this pathogen. That's why this mechanism is extremely efficient. Okay? Is that clear? Really? Okay. If any questions, do not hesitate. So now the natural killer cells. I said that natural killer cells are one type of lymphocytes that are not specific of the antigen. They develop in the bone marrow. They are called large granular lymphocytes because of their morphology, as I said. They have no antigen-specific receptor. No. They kill target cells by direct contact and release of perforin, so enzyme that will drive holes in the membrane of the cell, and granzymes, enzyme that will digest component of the cell. The NK cell kill mainly viral infected cells and tumoral cells. So they are a very important mechanism of surveillance for cancer cells. Okay? Now, how do they work? They do not have a receptor that is specific of a pathogen, but they kill some pathogen. Why? Because they have this system. As I told you yesterday, one of the mechanisms of evasion of the viruses is to lower the level of expression of MHC class 1. Why is it important for the virus, for the survival of the virus? Why? Because you will see it next time. CD8 T cells, the cytotoxic T cells, kill a cell which is infected by a virus when it presents with CMH class 1 molecule its antigenic peptide. Okay? So MHC class 1 molecule presence is very important to present the viral antigen. So the virus, in its genius of trying to evade the immune mechanism, it lowers MHC class 1 expression to avoid the specific CD8 T cytotoxicity. This is the idea. And you know that all our nucleated cells have MHC class 1s. So any cells that is infected by the virus have this mechanism of antigen presentation. So when it lowers the expression of MHC class 1, what happens? We have the second barrier, which is NK cells. You see here, the NK cells recognize directly, we said, the cells. When they find expression of MHC class 1 molecule at the surface of any of our cells, they say, stop, this is self, don't kill it. Okay? But when the virus, in its attempt to avoid the immune responses by CD8 T cytotoxic cells, lowers the expression of MHC class 1, it will avoid CD8 cytotoxicity, but NK cell will say, oh, this guy doesn't have MHC class 1, kill him. And this is the way it works. It's not specific in the sense that we have a receptor to recognize the antigen, but it is indirectly extremely efficient. So you see how nature have evolved. It's, as I said yesterday, it's an interplay between the pathogen. Who will survive? That's the whole story. Okay? So these are mechanisms that let us avoid this. So once this activation happens, we have two types of molecule, I said. We have the perforins, which make these pores in the target cell. This is the target cell, and this is the NK cells. And we have liberation of these granules that are enzymes, like enzymes. And all of this with the cell death mechanism, because there is all our cells have the program for cell death. Okay, normally all our cells. So this will be another signal. So we have death cells and all these perforin and enzymes to kill the target cell. Okay, is this clear? Really? If any questions, do not hesitate. All right. Now, I've told you we have this cellular component of innate immunity, but we also have a humoral component. When you say humoral, means that these are proteins generally, which are circulating in the humors of the organism, blood mainly, but also in the interstitia, in cephalorachidine liquid, for example, in all our synovial tissues, etc. So these are soluble factors this time. And among the soluble factors that are involved in innate immunity, the major one is the complement. What is the complement? We call it complement because it complements the actions of immunoglobulins. It helps the immunoglobulins to achieve their objective. Okay, that's the starting point. It has been called like this by Landsteiner long time ago because of this reason. So it is not one protein, it's a system protein. It's 20 proteins approximately. We have proenzymes. So like you know the coagulation, the hemostasis system. You remember you've got some teaching about that or not? No? Okay, it's similar to that. So we have proenzymes that will be activated one after the other. You activate one enzyme, you produce produce different component of one element, and then you produce a new enzyme, and etc. This is the way. It's a cascade. I was looking for the word. It's a cascade of events like this. Okay, so it's 20 proteins found in the plasma, so circulating. Most are proenzyme. I've just said do not have yet enzymatic activity. They get their enzymatic activity once the activation starts. Okay, then they react in the sequential cascade fashion like a clotting factors, what I've just said, and major participant in natural defenses of the organism. Two major activation pathways are recognized. One we call it the classical pathway, and one we call it the alternative pathway. And I will show you these different pathways. So the classical pathways is also approximately the same than lectin pathways, which is more recently described. Both of them use the same sequence mainly. So, but for the classical pathway, it's an immune complex that start things. When you have the bacteria which enter the body, you have antibodies that recognize the bacteria. And bacteria plus antibodies are immune complex. Immune complex is antigen plus antibody. Okay? So you have formation of immunocomplex. So the bacteria is there, and you start activation of the classical pathway of the complement system. It's a cascade of events, as I said, several components. And alternatively, there's another pathway which uses the antigen that are present at the surface of the pathogen. It does not go through the immune complex and the presence of the immunoglobulin. It's immediately, you have the antigen at the surface of the pathogen, and you will start this alternative pathway. Similarly, using different components like you see here, this cascade will lead, both cascades will lead to the production of the C3, and the C3 molecule is cleaved by different enzymatic activity, and will give two types of components. One type are the C3A. I see it in the next one, I think. So this is the classical, this is the alternative, and in the next slide, so we see no, I don't see it here, sorry. let me go back here. So this C3, when it is cleaved in two components, we have the C3A, which is an anaphylatoxin, which favor the inflammation. C3A and C5A are two components that are anaphylatoxin, and they will create an inflammatory process. They will start an inflammatory process. But you will also have the C3B, which is component of the two pathways, and will lead to the production, of this lytic pathway. So we have C5b, which will interact with C6, C7, C8, and lead to the formation of C9, the last component of the complement, which is the membrane attack complex, which is like the perforin I have just described. They will create a hole within the target cell, which is a bacteria. And when you create a hole, there is an exchange, an osmotic exchange, and the bacteria will be lysed. Okay? Lysis of the bacteria. All right? So please remember these two aspects of complement. One, it helps the inflammation with C3A and C5A. Second, it will be lytic for the bacteria or the pathogen that is there. Okay? Then that could happen with our own cells sometimes. That's why the complement needs to be controlled strongly. It's heavily controlled by inhibitor that will limit the activation, because an excessive activation of the complement could be very damaging, including to our tissues. Okay, now this is the way so it works, and I won't stay much more on that. So we have different steps, as I said. The classical pathway, the alternative pathway. The classical one is activated by antigen-antibody complex. The alternative pathway is activated by bacterial and viral surfaces. And two different ways at the beginning, but then they join one or the other, and they will lead to the lytic complement. Okay, so these are the actions of the complement. So it will lead to lysis of the bacteria by the mechanism we just described. It will help the inflammation with the chemotaxis. It helps the chemotaxis, so the mobility of the inflammatory cells to the site of infection. And it could be indirectly helpful in opsonization. Remember, I said it could be through fragment of the complement which are adhering to the bacteria. Okay? So these are the mechanisms by which the complement functions. And these are a picture of the lysis, as you can see here. You see it by electronic microscopy. You see the hole within the target cells. All right? So now I will go rapidly to the last aspect, which are interferons. So chemotaxis I have described. Other cells then, of course, phagocytes could interfere in this. Mast cells and basophil could help in this inflammation, of course, and other white blood cells and even platelets. All right? So opsonization. Now the last aspect I would like to talk about rapidly are interferons. So interferons are produced by type 1 interferons. There are different types of interferons. I'm concentrating on type 1, which are interferon alpha, beta, and gamma. Gamma is an immune interferon, but alpha and beta plays a role against viruses. So when a cell is infected by a virus, it will produce interferon alpha and beta, and these interferons will protect the other cells that are around against the viral infections. We can see it rapidly here. You see, this is a cell which is infected by the virus. The virus enters, uses the machinery to produce its own component, and this will lead to the production by the cell of interferon. This interferon is important to increase NK activity, remember, against viruses, and the expression of MHC products. So starting the activation of the immune system. So interferon will be produced also and with this aim, but also with by protecting the surrounding cells, which are around the cell which is infected, and this by enhancing its protein synthesis and leading to an antiviral state, better immunity against this virus that is around. Okay, so it's an indirect effect. The cell is infected, produces type 1 interferon 1, and those interferons will play two roles: one, activating the immune system, and two, protecting the contiguous cells, the cell that is close to it, by this better activity and better antiviral state. So those are the different components of the immune system, some of the innate immune system. Some are also important, which are proteins produced during the acute phase of the infection. And you certainly all have heard about CRP protein, the C-reactive protein, which is a marker of infection and inflammation that is used commonly in clinical. And these are produced very rapidly at the first steps of the infection, and they are important to promote phagocytosis, to bind to bacterial phospholipid, and to activate the complement. So and there are several others that are produced by the liver in the what we call the early phase of infection, the acute phase of infection. Okay? Is that okay? Any questions? Hi, Jamil. Questions? No? Okay. If any questions, I will be available, of course, at the department at any time.